All right, so chapter 13 then is about the specific defenses or acquired defenses. And I said in the last video, this is truly the immune system. So what we're going to see in this chapter is that we have cells in our body that their whole job is to run surveillance of the body and be looking for those foreigners. Again, key in this system is that ability to distinguish self from non-self, right? We need to know when, with this system, it's very powerful and it can destroy these foreign microbes. We don't want it to destroy our own cells, right? So if we do end up with lymphocytes that are attacking our own cells, that would be considered an autoimmune disease. So that's what we're trying to avoid here. And so hopefully, if the system is working right, we are identifying, attacking, and destroying these foreign invaders. So again, that first flow chart kind of talks about this. It's like this totally separate branch, right? It's truly immunity, and it's really that third line of defense. Remember, our first line of defense was intact skin and mucous membranes, and then the second line of defense had to do with things like inflammation and phagocytic cells. So this is the third line of defense. The key cells, the cells that work in this system are the lymphocytes. Now remember, all of our white blood cells together are called leukocytes, but just the B lymphocyte and T lymphocyte are the lymphocytes. So keep lymphocytes and leukocytes straight there. Lymphocytes are unique in that they have a special protein on their surface that's called an antigen receptor. So an antigen is really a protein that can stimulate an immune response. And so if an antigen stimulates an immune response, it does that by its interaction with these surface proteins on lymphocytes called antigen receptors. So the antigen receptors are a key to a lot of this. Now, this is figure 13.1. This is kind of a really nice overview of the whole system. So let's run through this and then we'll jump into some details. So remember the lymphocytes, those were coming from that lymphoid stem cell. This is a totally different lineage of cells than that made the rest of the blood cells. And so some of these lymphocytes are going to remain, excuse me, these lymphocytes are in the bone marrow. Some of these lymphocytes are going to remain in the bone marrow to mature, and those are going to become the B cells. So B cell, B lymphocyte, same thing. Once they've matured, they can move out into things like lymph nodes and the spleen. So parts of that lymphatic system, right? The lymphatic system is in charge of kind of maintaining these lymphocytes. That's why the names are the same. So again, in lymph nodes or the spleen, some of those tissues where they're going to hang out and be able to screen either the lymph or the blood. Let's just go with the B lymphocyte. So what we're going to see is that B lymphocytes, to become activated, they are going to need to be encouraged by T cells. But when they're activated, what we'll see is that some of the B cells turn into what is called a plasma cell. This is just a fancy B lymphocytes capable of producing antibodies. And so those antibodies are basically free proteins. They look just like the antigen receptor, but those are now going to circulate in the blood. And when they find the antigen, which is on the microbe, the pathogen, then they can attack it. So this, and again, we'll go into a lot of these details, but this whole side, this B cell lineage, you'll often see people talk about what's called humoral immunity. So the humors, that's like a old-fashioned way of saying like the fluids of the body. And so here we see antibodies going into the plasma, into the fluids, right, and circulating. So B cells are associated with humoral immunity. Now, the T cell lineage, the T lymphocytes act a little differently. The stem cell was in the bone marrow, but then the immature T cells are going to move into the thymus where they mature. So the thymus has this really important role of kind of screening the T cells to make sure that we only allow those that don't attack our own cells, right? So that self versus non-self, a lot of that screening takes place here in the thymus. Again, then, once they've matured, they'll move out into the lymph nodes and the spleen. Now, the T cells, we're going to have two different well, there's actually more than two, but the two we're really going to talk about. There are cytotoxic, so here we go, down here they're showing them. Cytotoxic T cells, cyto means cell, toxic is toxic. And so these are the cells that are actually going to be toxic to other cells. If they find a virally infected cell, they're going to kill it. Right, so that's what a cytotoxic T cell does. Helper T cells, you've heard because of HIV, and the helper T cells are really going to be the ones that kind of direct the immune system. They're going to do that activation of B cells. They're also going to activate cytotoxic T cells. Okay, so that's kind of the general thing. One other thing to note, and we'll see this in both lineages, we develop what are called memory cells. Memory cells are the key to immunity. So when you go ahead and make activated B cells and turn into plasma cells to produce antibodies, you tuck a few away in your back pocket for the next time you become exposed to this pathogen, right? Same with the T cells. All these classes of T cells, you leave a few behind so that when you're exposed to the same disease more than once, you can have a better response the second time. This is what we call immunity, right? That you really should only get sick from any particular illness once, because any other time you're exposed, those memory cells should hop in and take care of the pathogen before it makes you sick, right? So that is the overview of what's going on here in our acquired or specific immunity. So let's jump in and take a look at a few of these things a little bit closer. So I mentioned that both of our lymphocytes have these proteins on their surfaces called antigen receptors. And again, the antigen receptor is what's going to match an antigen and basically signal to us that this is a foreign molecule, not one of our own molecules, right? And really kind of kick off the immune response. So the crazy thing with these antigen receptors is that you make millions of different shapes of antigen receptors. Every lymphocyte only has one shape, right, over the entire cell. They're all identical. And so what happens is during the development of these lymphocytes or during the production of these lymphocytes, what happens, so this is what an antigen receptor looks like. So when I draw them, I make them U's and boxes and Y's, but really they all kind of have this Y arrangement. And parts of this protein, right, this is going to be a protein on the cell surface, are really identical, right? This is what it means to be an antibody. But then the ends that are actually going to interact with antigen, it's called the variable region. And so what happens is when you're building lymphocytes, you randomly select, that's what they're trying to show you here, different combinations, right, of the DNA so that you get slightly different amino acids, slightly different, and therefore slightly different proteins, slightly different antigen receptors on the surface. And what this does for us, right, as long as you have a diverse set of genes in this part of your genome, you make so many different antigen receptors that you're pretty well covered, right, as far as disease goes. And I will say you don't make these based on what diseases are. You just make a ton of them, right, and hope that you're going to find the right things. And so we do. We make antigen receptors for things that don't even exist, like Martian dust or, and it works really well because we actually end up having receptors that fit pretty much everything. Okay, so this is like a little nerdier version of that same picture, but again, showing that the variable region is what changes. And again, this is due to kind of a genetic recombination. So each lymphocyte is going to look very different. Now, so you make a lymphocyte and it has this unique antigen receptor right all over the surface. And so here we're seeing, right, all of our different lymphocytes. Notice they're doing different shaped receptors. And this picture is trying to show this idea that when the thymus screens these cells, any of them that actually match one of our own cells, right? A protein on our own surface matches it. Notice that is going to be told to undergo apoptosis, right? It's going to get taken out. We don't want that around. And so what's actually released into the blood, right, is a set of lymphocytes. There literally might only be one lymphocyte with this receptor shape, right? And one lymphocyte with this receptor shape. And so what happens is that first time you come in contact with the antigen, say it's a protein on H1N1, right? Or a protein on COVID, you only have one lymphocyte that recognizes it. But notice what happens. These cells are going to undergo what's called clonal selection, right? So it's like you have all your lymphocytes out in the blood, and we have this new pathogen, right? It's in there. We have a lymphocyte that matches, and it's like, you are the chosen one, right? Like, clone yourself. We need more. And so that's exactly what you're seeing down here, right? Is one cell becomes two, becomes four, right? And so you hit this massive, what we call clonal expansion of that selected lymphocyte. That's the one that we need in order to do the job. Okay, so let's see. So we're going to start on the T cell side of things. So a lot of little specific chemicals, specific proteins that make a big difference here. So hold on tight, pay attention here. Okay, so in this case here we have a microbe being engulfed by a macrophage or a dendritic cell, right? Something like that. That macrophage, once it engulfs that microbe, remember the phagolysosome is going to tear that up. It converts to what's called an antigen presenting cell. It's still the macrophage, right? But now a big part of its job is going to be to hold a piece of that microbe or many pieces of that microbe on its surface to show the rest of the immune system, right? Like macrophages weren't cool enough, just phagocytizing all these, foreigners. Now it's actually going to process them and stick them on the surface. So the way it does this is antigen presenting cells have this special protein called MHC2. This is major histocompatibility complex 2. This is a protein that the cell makes, right? So you have to picture a ribosome reading a little stretch of messenger RNA, building the polypeptide chain for major histocompatibility complex 2. But then it's going to be a surface protein. So we're going to send it through the endoplasmic reticulum and then the Golgi apparatus into the surface. And while it's in there, right, while it's been built, it's around all these other proteins, right? And so what it does is actually takes a sample of the protein that was in that phagolysosome, right, and carries it to the surface, right? And so here we see the antigen being held on the surface. And now we're going to zoom in closely. So this is our antigen presenting cell. The big brown corn dogs here are the MHC2, and they hold the antigen out, all right? Okay, now a helper T cell is going to be who checks antigen presenting cells. So the helper T cell has a special protein. I'm like, that also looks like a corn dog. A special protein here, this brown one sticking up, called the CD4 protein. So sometimes helper T cells are called CD4 cells because they have this protein that basically is able to dock, right, on the MHC2. So CD4 proteins dock on MHC2, which means helper T cells are always checking antigen presenting cells. Now, when the correct helper T cell, right, the one here in green, this kind of green clover looking thing, is the antigen receptor on our helper T cell. So when the correct helper T cell that has the right antigen receptor locks on there and says, aha, that fits, it must not be us, then the immune system, right, is starting to work, right? So one of the big things that helper T cells are going to do, right, it becomes activated. It's going to release different chemicals. The one they're pointing out here is called interleukin, and this is going to be a growth factor. So this is going to tell helper T cells, cytotoxic T cells to be activated, right? And they're also going to activate B cells as well. Okay, so that is antigen presentation to a helper T cell. Now, here's our CD4 cell that's just been activated. Okay, and it goes into a lot of detail, and we're not going to do all of this, right? But notice some of them, we've selected the T cell with the correct receptor shape, so it's going to clone itself, okay? And they're just showing us a small sample here, but some of those clones are going to become memory helper T cells, right? We're going to save those for later. They're pointing out a few that are producing things like the interleukins, right? And so we see that they're using chemicals to activate B cells, right? Make them become plasma cells, produce antibodies, make them create memory cells. Okay. We also see that helper T cells are able to do things like stimulate macrophages. We also, and so there's so many different helper T cells here. These stimulate inflammation. These are, I think, called repressor T cells, T sub R. And those are going to be ones that actually say, okay, it's over, the fight's over, we won, like, chill out. Also helpful for preventing autoimmune disorders. Okay, so then this lower picture, what we're seeing here is how a CD8 or cytotoxic T cell works. Okay, so let's see, where should we jump in? So here's our helper T cell, okay, and it's going to activate a cytotoxic T cell that has the same receptor shape, right? That's going to be the one that can match the microbe. So our cytotoxic T cells, some are going to clone themselves the same way. Some are going to remain memory cells, and some become activated. And when we say activated, what we mean here is cytotoxic T cells, their job is to check all the nucleated cells in the body, right? So they're not just checking antigen presenting cells like helper T cells were. Cytotoxic T cells check every cell in the body. And the way they do that, let's see if I have it zoomed in. I want to zoomed in. The way they do that is all the cells in your body have an MHC1 protein on their surface. And it works very similarly. It's produced by the cell, and it brings sample of proteins that are being produced in the cell to the surface. Now, if this cell is healthy, every protein that it brings to its surface should be one of your own. Like, look, I know how to make this. Look, I know how to make this, right? And the cytotoxic T cells don't care. They're like, cool, you're one of us, right? And they move on. But if that cell, say, has a viral infection, some of the proteins that are MHC1 are going to carry to the surface is going to be like a viral spike or a capsid or something, capsomere, something like that. And that's going to be our signal that a virus is hiding inside, right? Or if you have a mutation in the DNA of your cell, and so you start making funky proteins before that cell can spread and become cancer, the cytotoxic T cell should recognize that protein isn't right. This isn't us. Okay. And the way they do that, right, is if the self cell has MHC1 holding that protein, that antigen on the surface, what we're going to see happen is cytotoxic T cells have a CD8 arm, CD8. They're sometimes called CD8 cells. They have a CD8 protein that lines up with the MHC1 to dock. And then if their antigen receptor matches one of the proteins that's being shown, whoa, that means it's foreign, right? Because we shouldn't have antigen receptors to self proteins, okay? And so if it docks, if there's a match, the cytotoxic T cell has been activated, right, by helper T cells, and it's going to secrete fun things like perforins, right, poke little holes in this infected cell. and get it to lyse, get it to be destroyed. Look at that picture closely. I'm sorry, I can't zoom in, okay? But it looks an awful lot like this one, right? The difference being antigen-presenting cells have MHC2 and are checked by helper T cells, and then cytotoxic T cells have MHC, or check MHC1 with their CD8 proteins. Okay, so that's the T cell side. I can say maybe that's why I have this picture again. That's the T cell side of things, right? Activated T cells, some of them become memory cells. Helper T cells are telling everyone what to do. Cytotoxic T cells are running around and killing virally infected cells. Okay, so then let's see what happens on the B cell side of things. So B cells, they do have to be activated by helper T cells. And so that's what we see going on here, right? Our helper T cell, it's come in contact with the microbe, right? It knows what the correct receptor shape is. It's going to dock with a B cell. And what are they trying to show you there? So this is the chosen. They've already, okay, here's the microbe. And they're already telling us this is the chosen one, right? This is the one with the correct receptor shape. And so B cells also use MHC2. So B cells can be antigen presenting cells. And so it's going to hold a sample of that pathogen's protein on its surface, right? And docks with the helper T cell. And the helper T cell is like, yes, you are the chosen one. Go clone, right? And so again, that's the role of things like interleukins, these growth factors, to tell this B cell, right, to divide, okay? And so again, some of those B cells are going to turn into what are called plasma cells. Plasma cells tend to be larger, and they are just protein machines, right? Their whole job is to produce antibodies. The other name for antibodies are immunoglobulins, and we will look at what antibodies do specifically, right? Notice also we make memory cells for the next time. And then this regulatory B cell, we're not really going to go into. But again, there's always these lymphocytes at the end of things that are saying, okay, like let's rein that in. I think we got this taken care of. Okay, so this is the B cell side of things. So here's the antibodies. So the antibodies are proteins, and they look just like the antigen receptor. And because they're the same exact shape of the antigen receptor of the chosen one, right, they are going to be able to dock to the antigen on our pathogen. So a few different things they can do. So here we see antibodies just coating the outside of this bacterium. So you picture each of these ends attached to that antigen, to a surface protein on the bacteria. Now, that protein on the bacteria was there for a reason, right? Maybe it was a receptor, maybe it was an enzyme, and now we're blocking all of them, right? So you can start interfering with kind of the natural normal function of something like a bacteria, right? And that's going to be able to slow it down. The other thing that happens when this bacteria is totally coated in antibodies, it actually makes it easier for the macrophage to engulf it. So this is a process called opsonization, where it's almost like putting little studs on the outside of our bacteria so that when the macrophage tries to engulf it, it's easier to hold on. I like to think of it as like there's a pool game where you like grease a watermelon and then you're trying to like catch the watermelon, but it keeps squirting out. Okay, so if you had I don't know, stuck a bunch of like pencils in it, then you could grab onto it, right? And so it's like that for the macrophage. That's opsonization. All right, neutralization. Again, this is this idea that if you can kind of cover important proteins, right? In this case, they're showing antibodies attaching to a virus. And think about COVID-19, right? It has that spike that needs to dock on a receptor to even get into a new cell. Well, now we've neutralized this virus because the antibodies are covering the spike. It can't dock. It can't gain entry, right? We can also have antibodies cause this process called agglutination, where you start sticking a bunch of the microbes together. They're going to form these large clumps. It makes them immobile. It makes it way easier for a macrophage to grab a whole bunch of them. It's harder for them to like circulate through the body, right? So that's useful. It also acts as a marker for complement. Remember complement from the innate immune system? So they serve as like a little tag. Complement goes ahead and builds that membrane attack complex and blows them up. And then this one they're pointing out, so some bacteria, or I guess microbes, bacteria are producing toxins, and some antibodies can actually see that toxin as the antigen, right, as the foreign protein, and by blocking that toxin, it serves as an antitoxin, right? It can prevent the toxicity. Anyway, so what I love about this, right, is like, we're always like, oh yeah, the B cells make antibodies. And it's like, the antibodies are real and they do real things, right? So think about these mechanisms, like the way antibodies actually work. And I think that's actually way more important than knowing like all the ins and outs of these different types. But I do want to show you this, right? So there's 5 different classes of antibodies. We've got IgG, IgA, IgM. Look at this crazy guy. IgD and IgE. So let's pull out just a couple key things about each of these. So I would point out, right, IgG is the most common antibody. And then there's fun stuff like it lasts for 23 days. That's the half-life. So half of it is gone in 23 days. Anyway, it crosses the placenta. It calls in complement, right? All these different things. This is, I was going to grab this. Yep, so highest concentration, longest lasting, and is able to cross the placenta. That's what I wanted to point out there. IgA, right, notice it has kind of 2 ends on it. They're calling it a dimer. Again, not essential. But notice it doesn't cross, so I just think this is fun. It doesn't cross the placenta, but it actually does get passed on to a baby through breast milk. IgA is going to be really the main antibody on mucous membranes, right? So this is an important one for protecting, say, like the respiratory tract. IgM is this big crazy one. It's a pentamer. It's the first to appear. So following infection, you'll see IgM levels spike, but it does not last very long. IgD, fairly small amounts of it, and I would say it really has an uncertain role, but IgD does exist. IgE is another super important one. This is going to be, right, it's pretty uncommon, right? Very small amounts, but sorry, I'm in the way here. IgE, very small amounts, but it's really important in allergies and parasitic infections. So IgE binds to mast cells, basophils, and gets them to release histamines, right? So it's that trigger for the inflammation associated with things like allergies, okay? So again, it's good to know like a couple things about each of these, but it's more important to know what antibodies do. This is cool. So notice what happens here. So let me get the pen back, pointer back. Okay, so this is our first exposure to the antigen, right? So you get stung by a honeybee. Okay, no, that's not a good one. You contract H1N1, swine flu. Yeah, it's around, not a big deal. Okay, so you get exposed to it. Notice you have this latent period. This is the period of time between your exposure and when we actually start mounting our immune response. Now, this is going to take a while. And it takes a while because H1N1, I mean, what? You got a couple hundred viral particles in you. It's going to take them a while to replicate, move into new cells, replicate some more. before your immune system can catch on. And then your immune system is playing catch up, right? Because once it finally notices it's there, it has to go through that clonal selection, you're the chosen one, and then clonal expansion, right? Making all these copies, making plasma cells, and then making antibodies. And now we start to see antibodies rise. Again, notice IgM is the first one to spike, but then it kind of goes away. IgG takes a little longer, right, to have the specific IgG for this disease, H1N1, right? But then it goes up. And so these antibodies, agglutinating, opsonizing, neutralizing, all those things, our infection goes away, right? We recover, we get better. But you got sick in the meantime. Now, next time, right, a few years later, maybe you get exposed to H1N1 again. Notice, right, here's our exposure, here's our antibody production. What happened to the latent period? What happened to the lag time? It's gone. Memory cells were ready to go like that, right? Mount an immune response and just, man, look at IgG off, out of the blocks, just boom, right? And so we're going to get this. Notice this is a much larger production of antibodies and obviously much quicker. That's immunity, right? Where you can mount the response so quickly that you don't even get sick. Okay, so that is and that's also why they call it an acquired immunity, right? It doesn't just happen. You have to go through a primary exposure, right, in order to get to the point where you have those memory cells and where you have immunity. Okay. We already talked about that. These are just pictures from another book, also explaining kind of how helper T cells work, B cells. This I pulled from another book, and I do like it because it kind of shows the same thing that we just saw in that antibody graph. So here, this is our first exposure. This is our second exposure. That's kind of what this line is here. So notice what happens. Our first exposure, our antigen is engulfed. So we can kind of run through the whole spiel here. Engulfed by a macrophage, which becomes an antigen-presenting cell, right, and uses its MHC2 to hold that antigen out on the surface, and the helper T cell is like, ooh, I recognize that, right? Notice the helper T cell stimulates cytotoxic T cells and stimulates B cells. Okay, now once they're stimulated, right, and we select the correct B cell, it's going to go through clonal selection, clonal expansion, produce plasma cells and then antibodies, right, to fight off the disease. The cytotoxic T cells are going to have to go through clonal selection, clonal expansion, have these active cytotoxic T cell go around the body and destroy cells, okay? And so by the time you get to the bottom of this page, we've cleared the disease. But that took a while, took a whole page. You got sick in the meantime. Now, second time you come across that antigen, notice how much shorter everything is, right? Okay, so we're able to very quickly stimulate helper T cells, memory helper T cells, memory cytotoxic T cells, memory B cells, right? And it's like this direct exposure to the antigen, right? They're ready to go. They have little antigen receptors. If they bump into it, they're like, wait a second, right? And so here we go. We're active, we're killing, we're active, we're antibodying, boom. No sickness, just wiping things out. Okay, so that's how that works. Now, there's 4 types of acquired immunity. The classic example, kind of the way we've been explaining this, is what's called natural active immunity, right? And this is where you have to go through getting sick and the whole process of building those memory cells so that your second exposure, subsequent exposures, you don't get sick because you have memory, you have immunity, okay? But natural active immunity requires recovery from an infectious disease. We've circumvented things and said, well, this doesn't look like so much fun. Can we get that same active immunity artificially? Well, yes, we can. That's called vaccination, right? And so basically vaccination is just, there's different ways to do it. and you'll do some research on that. There's different ways to do it, but you're exposing someone essentially to an antigen, whether that's a dead microbe, just a protein from that microbe, etc. But what you're trying to do is get the immune system to mount the same response to an antigen that's not able to make you ill, right? Because it's been removed from those like pathogenic qualities of the microbe. This is super cool, like medical miracle, right? We've eradicated smallpox. We've, almost eradicated polio. We'll talk about more about vaccines, right? Okay. Other ways to get acquired immunity. So here's another natural example where you get immunity passively. Natural passive immunity is when antibodies either cross the placenta or cross via breast milk into the baby. And so this is basically handing the baby preformed antibodies so that if they become exposed to, whatever's going around, so say mom caught H1N1, right? She's giving the baby antibodies so that it's not going to get sick. It doesn't, how do I say this? So the baby is basically protected for somewhere between like a few months and a year, depending kind of on what the antibodies are, how many antibodies mom had circulating, what disease, that kind of thing. But this is cool. It's handing off, right, that immunity before it's actually acquired immunity. Okay. And down here, they are pointing out the infant's getting 99% of its natural passive immunity in utero. Most of it is from crossing the placenta. Okay, so then we thought, well, wait a second. If a baby can get antibodies from its mom, is there a way we could do that artificially? So here we go, another form of artificial immunity. This is a form of immunotherapy where a patient is really receiving preformed antibodies. So you'll see this sometimes. So we've seen this with COVID, right? They're talking about people who've recovered donating plasma. The antibodies circulate in the plasma. And so the idea here is to harvest those antibodies and give them to someone else who's sick so that those antibodies can agglutinate and neutralize and opsonize, right? All those things before their B cells are even making those antibodies, right? And so it's a cheat. It's giving your body a head start on the pathogen. And it's been really successful in some cases, right? It's, best of my knowledge, like the only treatment we came up with for things like Ebola. You can also get what are called monoclonal antibodies. So you could say with like rabies, right? You can infect a rabbit with rabies and then collect antibodies from the rabbit. You could also give the gene for how to make those antibodies to something like a bacteria and get it to produce the same proteins, the same antibodies. So that's actually a really neat kind of therapeutic. And so they're looking at that with COVID. And I know President Trump did get, I think he got monoclonal antibodies. So I do think that's something that we will be hearing more about here in the future. So vaccination. So one of the things you're doing this week is actually watching a PBS documentary called Vaccine Wars. So I don't want to go too far into this because you're going to be writing a little paper, but just keep in mind, right, the first at least acknowledged vaccine, 1796, Edward Jenner. The idea here is he basically, as the story goes, right, who knows how accurate it is, but as the story goes, right, smallpox is running rampant. Smallpox, of course, killed millions of people. And Edward Jenner notices that the milkmaids don't seem to get smallpox. They contract a closely related virus called cowpox. They get it from the cows, right? They're hanging out with the cows a lot. And so he starts thinking like, okay, what about that cowpox exposure is making them never get smallpox, right? Somehow they're immune to getting smallpox. And so, as the story goes, right, he basically scratches a pox, right? Oh, you guys might not have had chicken pox, which is good. Those like blistery things on your skin. He scratches that, right, and basically inoculates a boy, probably his own kid. That's how scientists did things, right? Like stab it into his own boy to see if that was what was conveying immunity. And there we go. We start vaccination. Now, I've actually heard of stories from much earlier than that, that it was widely used in China previously to that. And so there's a potential that Edward Jenner is getting the credit, but that other cultures already used similar techniques. Even in the early days, there were people who were scared of vaccines. So here we see, right? So cowpox could keep you from getting smallpox, but these people who were vaccinated, they're turning into cows, right? And I've already seen my mother-in-law was questioning, right, this video she saw because a doctor says you shouldn't get the coronavirus vaccine because it's human 2.0. They're genetically modifying you, right? So it's like there is nothing new under the sun, right? Even back in the, 1800s, people were saying, I don't want to get that vaccine. I'm going to turn into, a cow. And then other people are lining up and saying, please, I need this vaccine. Like, I don't want my child to die. And so, yeah, we haven't moved that far. So, as you think about vaccines, in chapter 13, they do go into some more detail on how vaccines are made. I want to see kind of what you guys write about, what you found interesting, and then we can fill in some of the blanks there in class if someone hasn't written a paper about a specific, about adjuvants or things like that. We can kind of fill that in, but I'm going to leave it there. Look into some vaccines a little bit more. Again, you can write an opinion piece on the movie, but also feel free just to explore new vaccines, right? Or new technologies. It's one of the big things right now is they're trying to make, instead of a vaccine where like you'd give someone the protein and have their body respond to it, they're trying to make vaccines where they actually give you the DNA of the virus and you build the protein and then your body responds to it. So those are DNA vaccines, and that's a new technology that some groups are studying with coronavirus. So anyway, there's a lot of stuff out there. Find something that interests you, and we'll see you in class.